Okay, so today what we're going to do is we're going to dive into the plant cell and the bacteria cell. The plant cell is going to be very similar to the animal cell that we did the other day. So we're not going to have to, the good thing is, you know how we had to write a ton of information the other day for the animal cell about their functions? We're not going to be doing that today. We're just going to review it and go over that. Okay, so a plant cell, just like the animal cell, is eukaryotic. Okay, that means it has a nucleus. Alright, plant cell has a nucleus. Do we remember that little trick I told you? It was last week, right when we started this chapter, about how to remember eukaryotic and prokaryotic, which do and don't. Isaiah? Oh, will you raise your hand to tell me? Oh, you just remember it?
Yeah.
Okay. Kenzie, I saw you shake your head yes. Do you remember what it is?
Yeah. Pro means no.
Pro, like in prokaryote, means no nucleus, right?
Yeah.
And then, do you remember the other one?
And U means do.
Yeah, like in eukaryotes, means do. So eukaryotes do have a nucleus, prokaryotes have no nucleus. Okay? Alright, so plant cells, they have all of the same organelles as animal cells, except they don't have centrioles, flagella, cilia, and then they have new structures that animal cells don't have called chloroplast and a cell wall. Okay, so we're going to start out with our drawing just like we did for the animal cell by drawing the phospholipid bilayer. Okay, so we're just going to draw it little, not around the whole thing again, because that would take a while. So we've got our phosphate heads and our fatty acid tails. Okay, and then we have our integral protein. Do you remember what the function of the integral protein is? If you don't, look back in your notes. Savannah?
It's the door that the molecules in and out of the cell.
Yeah, it's the door that allows the bigger molecules to get in and out of the cell. Right, so we have our integral protein. All right, and then we have our peripheral protein on the outside here, and their function is that they connect cells together. All right, and then we have our carbohydrate out here. Do you remember what the function of the carbohydrate is? Kenzie? To do what?
Identify.
Yeah, identify cells and has communication. And then we have, this is where you want your other color just to kind of show, cholesterol. And then the function of cholesterol is to keep the fats from sticking together, and it allows with cell expansion. Alright, so that's the main parts of the phospholipid bilayer that we talked about, which makes up the cell membrane. Right? So we can draw that around. Going to make it kind of big again. Okay. Ooh, sorry. All right. So what the main function of the cell membrane, the plasma membrane, is to protect and regulate what comes in and out of the cell, right? But plant cells have this extra layer of protection, this extra protective barrier called the cell wall. So that is just an extra layer on the outside. All right. And the main function of that is protection of the cell. So the plant cell has extra protection compared to the animal cell. So we've got our cell wall, and then we've got our cell membrane. Oops, sorry. And remember, if you can't see something or if I forget to move it, because that will happen sometimes, I just get in the groove, just holler at me, okay? Alright, so, like I said, plant cells are very, very similar to animal cells. So what we're going to start with is just the cytoplasm. Okay? And what the cytoplasm does is it does this reaction called glycolysis that makes 4-ATP from sugar. Okay? So we've got the cytoplasm. The next thing we're going to draw is the nucleus. Alright? Do we remember what the nucleus contains? DNA in the form of what? It starts with a C. Chromatin, right. Chromatin's that bundled up DNA, right? And what else is in there? Starts with an N. Nucleolus, right. So the chromatin is the DNA and the nucleolus holds the RNA, right? What's on the outside of the nucleus that protects it?
Isn't that like the thing that's got, like the nucleus pores or whatever?
It does have nuclear pores in it, but what's it called? Kenzie, did you say it? The nuclear, starts with an E? Envelope, right, the nuclear envelope. Good job. So we've got our nuclear envelope that protects the nucleus, protects the information inside of the nucleus. And then there are the nuclear pores that are in it. That's what allows the RNA to exit, right? So we've got, let's label some stuff here, okay? So we said this was our nuclear envelope. These are our nuclear pores. Our nucleolus. It's a funny word, isn't it? Nucleolus. And then we've got our chromatin, which is DNA. We don't have to rewrite all the functions because you have them from the animal cell, right? So the chromatin is our DNA, the nucleolus holds our RNA. These nuclear pores in the nuclear envelope allow the RNA to exit, right? And this is just, that's our nucleus, we know that. Okay. We feel pretty confident with that so far? Give me a little feedback, guys. Doing okay?
Yeah.
Alright, cool. Cool. Alright. So, does anybody need an extra second to do this? Okay. So now we have the rough and smooth endoplasmic reticulum, right? So we have that coming off of the nucleus. What is on the rough endoplasmic reticulum, the rough ER that makes it rough? Kenzie? Ribosomes, right. So you can draw that in another color. So we have ribosomes. Alright. So we have rough ER and we have the smooth ER. Right. And these things are called ribosomes. The rough ER and the ribosomes both make proteins because both are made of RNA, and RNA is the protein maker from the DNA instructions. Okay? And then there's also like free floating ribosomes. What do we think their function is? You okay? Yeah. I didn't hear. Okay, I didn't hear you said. I thought you had a question. Okay. All right. All right. So we said the rough ER and the ribosomes make the proteins, and then the smooth ER, that's what makes fats and breaks down the toxins in our body. All right. The next thing we have, apparently I was saying this wrong. I said Golgi apparatus, right? It's a Golgi. Why did you guys not tell me I was saying it wrong? That's how I pronounced it my whole life, but then I listened to it on Google and it's Golgi. It's okay. Alright, so we have the Golgi apparatus. What's the function of the Golgi apparatus? Look back in your notes if you don't know. Yeah, it packages and ships the material out into the cell. That's like, it's our FedEx or a UPS of the cell. Alright, and then we have the vesicle at the end here. That is the package that the Golgi apparatus ships out. Okay, that is the package. So the next thing we're going to draw is the mitochondria. It's got this double membrane. Ooh, that's ugly. I'm sorry. It's got the double membrane. What is made in mitochondria? Yeah. ATP, right. Makes ATP from sugar. Mitochondria makes ATP. Okay. Alright. Then we have the lysosome. The lysosome. What's the function of the lysosome? Said it looks like the Pac-Man, Liz?
It cleans up all the stuff in the cell.
Yeah, it cleans up the cell, digests the waste, right. Lysosome. Remember when we played that game the other day? When we were the spaceship going into the cell and doing investigating and the lysosome was trying to kill us because we were an intruder. We were a foreign thing inside the cell. And the lysosome was like, nope, not okay. Trying to kill us. Right? That's what it's doing. It's trying to clean up the cell, get us out of there. Alright. The next thing we have are the microfilaments and the microtubules. We have microfilaments. And the micro. Right, all these things are the same. We saw all these in the animal cell. Alright, so the microfilaments, they are for structure and the microtubules are for transport. Right, none of this stuff is new. The only thing we've seen new so far is the cell wall. Alright. So, we need another second? Nope, we're good. Alright, so the next thing we're going to draw that we don't see in the animal cell though is the chloroplast. Okay, we've got the chloroplast. What are the things that we see inside of the chloroplast that we talked about? There are three things. One starts with a T, one starts with a G, one starts with an S. The stroma, right? What starts with a T? A T. Flip back. We talked about chloroplast on page 3. The thylakoid disc. And then what is a stack of thylakoid discs called? Grana. Yeah. Grana. Yeah. The grana. So, this is not going to be the prettiest. I'm no artist. Right, but we've got the stacks of discs. Stacks of discs. It kind of does. It's kind of ugly. I'm sorry.
Mine does.
Oh, mine's just a big snowman. All right, so.
That's a snowman?
No, it's not. All right, so one disc, that's the thylakoid. Thylakoid disc. And we said a stack of the discs is called the grana. And then the stroma is on the outside. Alright. And this overall is our chloroplast. What reaction occurs in the chloroplast? Photosynthesis, exactly. Good job. Right? So what the plant cell does is it uses light, water, and carbon dioxide in photosynthesis to make their own sugar, and oxygen, right? So it makes sugar and oxygen, and then that sugar and oxygen goes from the chloroplast into the mitochondria, and that's where the mitochondria uses that oxygen and sugar to make the ATP for the cell. What's ATP? Energy, right? That's the cell's energy, right? And then the last thing we have is a vacuole, but the one in the plant cell is way bigger compared to the animal cell. Got this big old vacuole, right? It holds what? Water. It holds water. And plants need a lot of water to do photosynthesis, so we need a way bigger vacuole. Okay? Alright. So yeah, very similar to the animal cell, right? We have this extra protective layer, the cell wall, and we don't have cilia, flagella, or centrioles. And we added a chloroplast, and our vacuole is just larger. Okay? So is everybody done writing down? Okay, so we already talked about this, but let's just write some stuff down. So cell wall, so that's the extra protective barrier. And its main function is to protect the cell. Main function equals cell protection. And the chloroplast, that's where photosynthesis occurs. What's up, Ben? We are doing bacteria cells today. Yep. We're finishing the notes up today, and then we'll have no questions. Okay?
Yay.
The bacteria cell is simple.
So if I do the questions, will that go
It will, yes. Alright, and then the vacuole we said, very large, holds water. Alright? How are we feeling right now? Alright, am I good to flip the page, or are we still writing? Alright. Okay. Alright, so now we're going to talk about bacteria cells. So bacteria cells are prokaryotic, which means they have no nucleus. Okay? No nucleus. Pro means no. So you're going to notice that bacteria cells are going to be fairly simple structure-wise and organelle-wise compared to the plant and animal. So we're adding some new things, though, that you'll see. Bacteria cells have 3 layers of protection. They have the cell membrane, they have the cell wall, and then they have this thing called a capsule, which is on the outside. Alright, that's just this, it's made out of protein, and it's this outer protection, right? So, if there are 3 layers, that means it's really hard to get into a bacteria cell, right? That means it's going to be very hard to kill. Like, has anybody ever had a bacterial infection? It takes a long time to kill the, yeah, Jordan? Think you might have? The bacteria is hard to kill because you got to get through 3 of those protective coverings, okay? So, we're going to draw the very outer one right now. It's very outer one. Okay, Okay, so let's. So this first one, this is called a capsule. It's called a capsule. So go down here to the functions. So for the capsule, what it is, it's a protective layer made of protein. Okay? Protective layer made of protein. Alright. So then we have our second protective layer. That is called the cell wall. Cell wall, like we just talked about in the plant cell. The cell wall, its function is protect the cell. Oh, sorry, protect the cell. And then our final, most inner layer of protection is called, what do we think this is? The cell what? Membrane, the cell membrane. The structure that animal, plant, and bacteria cells have. Cell membrane. Right? Cell membrane for protection, but it also regulates right? That's the phospholipid bilayer that is in phospholipid bilayer was in the cell membrane that contains the integral and peripheral protein, cholesterol, carbohydrates, phospholipids, all that good stuff, right? All right. So now that we've got, ooh, that's pretty ugly, and then I don't know how to draw straight lines. All right, but so now bacteria cells do not have a nucleus, but what they have is this thing called a plasmid or a ring of DNA. So what it is, all right. Okay, this is called a plasmid or a ring of DNA. You'll see it as both. Ring of DNA. And then we have ribosomes, like the plant and animal cell. And the ribosomes are the RNA for bacteria cells.
Do we not write anything into the
What's up?
Do we not write anything into the PI or I?
We haven't got there yet.
Oh.
Yep, we will, right? Yes, sir. Give me about another minute and a half. We'll get there. All right, and then we have the cytoplasm. And we talked about this a little bit when we talked about the animal cell. This is where, right, we said the process or the reaction glycolysis occurs here, right? So we have glycolysis, and that's where sugar is broken down into 4 ATP. Where is energy made? Where's ATP made in the plant and animal cell? Starts with an M? Mitochondria, right. But bacteria cells do not have a mitochondria. So this right here is where the energy is made for the bacteria cell, right? That is how it stays alive, because there's no mitochondria for it to make energy. Mitochondria makes 34 to 36 ATP from one sugar, but the cytoplasm only makes 4. So it's not nearly as much energy for the cell, but it's still energy, okay, which keeps the cell alive. All right, so the last thing, I guess, all right, so this last thing we need to talk about is, I say pili. I don't exactly know how to pronounce it. Pili, pili. I think it might be pili actually. Pili. Yeah, pili. Okay. So pili are these things that stick on the outside of the cell. Okay. Those little things that stick out on the sides. Okay. What? Yeah, they do a little bit. Okay. Their function is to stick into, like, are you two okay? Good question?
No, I said 4 legs.
They're not really legs.
It just made me think of it.
Oh, okay. Alright, we're almost done. Focus. So their function is that they stick, or like they adhere to things. So bacteria cells, they use asexual reproduction, which means a cell would split into make 2 cells. But sometimes bacteria will find other bacteria cells and connect their pili, and that they share DNA by passing their plasmids, and this mixes their DNA in order to create a better cell, right? When you combine the DNA, that potentially creates a better new DNA, creates better DNA, and that would potentially make the cells better. So what that is called is so the process. This is important to know. Process of exchanging DNA is conjugation. Okay? Conjugation. Underline it, star it, whatever you got to do, because we haven't talked about that before. Alright, so the pili allows the other bacteria to connect, and then that way they can share their DNA by passing their plasmids back and forth. Okay? So even though that these bacteria cells are pretty simple, they still have everything that a living thing needs to survive. They have protection. They actually have the most protection out of all 3 cells, right? They have a way to get energy. They have the protein recipe, and they have DNA and ribosomes. So they have everything they need in order to be a living thing. Okay? Do we have any questions right now? Okay. So go and get out your Chromebooks. Get on Canvas. We have Chapter 7, No Questions 3. All right? I'll be around to answer questions if you guys have any, okay?